Hello everyone, and welcome to this webinar, Tools to Advance CAR-T and TCR-T Cell Therapy Development, presented by Dr. Véronique Baron from BPS Bioscience. I'm Diana Perrier, Digital Marketing Manager at TB Bio, and I'll be your moderator for this session. We at TB Bio are excited to have you join us for this session. For more information on our proposed solutions, please visit us at tbbio.com. Before we get started, I wanted to go over the webinar console. I would like to remind everyone that you can engage with us by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Q&A box and click on the submit button. We'll answer as many questions as we have time for during the live Q&A session at the end of the presentation. Please take a moment to complete the survey that will appear in your browser when you leave the session. As you can imagine, your feedback is always invaluable to us. And if you lack more information, let us know. We'd love to follow up with you. Now, I'd like to introduce you to our speaker today. Véronique Baron, PhD HDR, is a cellular and molecular biologist with extensive research experience in drug discovery and development, target validation, and cancer cell biology. Dr. Baron has worked for governmental and not-for-profit institutions, as well as biotechnology companies. She has published numerous patents and scientific manuscripts, has trained scientists at all stages of their careers, and for many years sat on grant review panels for the DoD. She has been with BPS Bioscience for 4 years and is now the marketing lead. With that, I'll hand it over to you, Dr. Baron. Thank you again for joining us, and we hope you enjoy the session.
Hi, good afternoon, everyone. Today I will be presenting from San Diego, where it is morning for us, and I will be presenting some of our tools that BPS Bioscience has developed to advance research in CAR-T and TCR-T cell therapy. So, Diana, can you please give me control?
Normally, it's all good for you. You should be able to do it. Just a minute. Let's try again.
Yeah. Okay.
Perfect.
Yeah. So I will talk very briefly about BPS Bioscience and what the company does. Then I present some basics of adoptive cell therapy. I'll present on CAR-T cells, TCR-T cells. I have a couple slides on custom services that we offer for CAR and TCR-T cell development, and I will show just 2 posters that may be useful for you in this area. So BPS Bioscience advances new scientific discoveries that lead to therapy by creating innovative solutions for research. We were established in 2005 by Dr. Henry Zhu, PhD. As you can tell, this is our 20th anniversary this year. We're very proud of it. The company is very science-driven, with very highly skilled and knowledgeable teams of scientists. About 40% of our team members have PhDs, and we are always here to answer questions from customers. We design and manufacture our products here in San Diego, California, and we have a global reach with extensive offering of products and services to provide value worldwide. We started the company really by producing proteins and antibodies. Our proteins are highly purified extracellular proteins or recombinant enzymes with enzymatic activity. We developed assay kits to study all these proteins, and we now have an offering of over 600 kits. We offer virus-based tools, and we have an extensive portfolio in cell lines that can be used for cell-based assays. So I'm going to present a few slides of introduction of adoptive cell therapy. What are the principles of adoptive cell therapy, the challenges that are faced by companies that develop these type of drugs, and what would a typical workflow be when you're developing drugs, and how can we help you during that workflow? Cell therapy harnesses the body's own immune cells to target and kill unwanted cells such as tumor cells, rogue immune cells, or infected cells. So it really started with cancer when scientists were trying to target tumor cells through cell surface proteins that are expressed specifically at the surface of tumor cells. But it has now evolved to also include autoimmune diseases and infectious diseases. So for cancer, both CAR and TCR strategies are used. In the case of CAR, we're targeting tumor-specific cell surface proteins. Some examples that I've shown here are BCMA, CD19, CD20, and so forth. There are currently, I believe, 8 or 9 CAR-approved therapy, which are targeting either CD19 or BCMA. And then the field evolved with TCR strategies that are targeting tumor-specific peptides that are presented at the surface of tumor cells by the MHC complexes. So some examples of these tumor-specific peptides may be NY-ESO1, KRAS mutant peptides, MART1, MAGE, PRAME. FDA approved and there's one NY-ESO1 approved TCR-T cell and a PRAME T cell that is pending FDA approval, depending on clinical trial results. For autoimmune diseases, the strategy right now is based on CAR with targets that are expressed specifically at the surface of B cells, such as CD19 and CD20. And the goal with these is to eliminate B cells that are producing antibodies against the self. And then for infectious diseases, the strategy is mostly through TCR, where the TCR can target viral or bacterial peptides that are presented at the surface of infected cells. And examples of these are HIV and the flu. Adoptive cell therapy uses the body's own immune cells, or they can use allogenic cells that are coming from healthy donors. So the immune cells in this case are obtained by blood draw. You take blood from a patient or a healthy donor, and you isolate the T cells from this blood draw. And then in the lab, these T cells are engineered to either introduce a CAR construct or a TCR construct. So the CAR stands for chimeric antigen receptor. It is a complete recombinant protein that is made in the lab. The TCR is also going to be a recombinant protein, but the TCR is closer to an actual TCR that would be expressed on the surface of a T cell. So these cells are engineered most of the time through viral particles at this moment, very often lentiviruses. The CAR construct or the TCR construct is introduced into the T cell. The T cells are grown in the lab to be expanded and multiplied. And once you have obtained millions of T cells, you reinfuse them into the patient, and they're going to attack the unwanted cells. So they're going to attack, for example, the tumor cells and kill them. So there are a number of challenges during the development of these therapies. And because they are human immune cells, they have very specific set of challenges for developers. So one challenge is the evolving regulatory landscape with, because the field is still evolving, different regulations coming up as we discover new issues, for example. Because they're living cells, they can die, so they're difficult to work with. They're difficult to obtain, they're difficult to manipulate, and they're difficult to expand. They have a very complex set of characterization steps and functional validation steps before you can get into the clinic. And then at the level of clinical efficacy, there are issues with rejection and there are issues with exhaustion. So new generation of cells are being engineered to improve the quality of the in vivo expansion, to improve persistence when they have been re-injected into patients, and to improve function, efficacy, and safety. This is a typical workflow for evaluation and validation of these new drugs. And this is at an early phase of evaluation and validation. We're talking here about all the experiments that are done in the lab, whether it's in vitro or in animals, but it's like the pre-IND phase, the pre-regulatory phase, and of course, before anything clinical is being done. So the first step would be to engineer the desired CAR gene or TCR gene. The slide was made for CAR, but TCR is very similar. So we can help here with PCR plus assembly kits that make it very easy to assemble your different pieces of DNA. You create your viral vectors, for which we offer lentiviruses and AAVs. These viral vectors are used to transduce cells to introduce the CAR or the TCR of interest. And for all these steps, we offer cell line, primary cells, media activating antibodies, and so forth. Then there is a step of target binding verification, verification of CAR expression, verification of TCR expression. And for these, we offer recombinant proteins and antibodies. Once you know that your CAR of interest or your TCR is expressed onto your cell, you can go through cell activation assays with cytokine assay kits that measure how cytokines are produced after activation of your cells. We have CAR luciferase reporter cells that are used as controls, one-step luciferase assay systems and media. And then the cell killing assays, which are usually co-culture assay, we provide cancer cell lines that express the target of interest. We have antigen overexpressing cell lines, luciferase reported target cells. So I'm going to walk you through a couple of case studies where someone is trying to develop, for example, a CAR cell or a TCR cell, and is at different stages of development. So we're going to see, for example, what a chimeric antigen receptor is. And in the case study, this person would be assessing CAR activity in a cell-based reporter system. We'll talk about recipient cells, target cells. In another case study, the scientist is now measuring cell cytotoxicity, which is more of a functional validation of their CAR T cell. So what is a chimeric antigen receptor? It's a construct that's usually made of an extracellular domain that recognizes the antigen. So it's usually part of an antibody, an scFv, for example, that is fused with a transmembrane domain, as you can see here. I don't know if I could, so I can get a pointer, but hopefully you see the arrow here. And then it is fused with an intracellular signaling domain that activates signals inside the cell that will activate the T cell for proliferation and for function. So the first generation just had one CD3 epsilon signaling domain, and then a co-stimulatory domain was added for the second generation. The third generation CAR constructs usually contain several intracellular domains that are meant to better activate the CAR construct and better activate signals inside the T cell. And the fourth generation and fifth generation are adding different elements, for example, an IL-12 inducer that promotes better cytokine activation, or IL-2 that promotes better cellular proliferation and better expansion of the clone of the T cells. So the field is still evolving. CARs are now engineered to address issues such as cell exhaustion, toxicity, activity, and the different scientists who are developing CAR will try different things. So let's get in the case of someone who's developing new CAR constructs, and they are optimizing both the transmembrane domain and the intracellular domain. They're trying to improve T cell activation. So what they would want to do is test all of these CAR constructs in parallel, see which one transmembrane domain allows for the best activation of the CAR construct, and which one of the signaling domain, for example, also leads to the best activation of the CAR construct. So they do have all their construct, and these scientists want a validated cell-based assay that would have a positive control so that they can compare the activation of different constructs. And because they want to compare them, this assay must be quantitative. So in that particular case, BPS Bioscience would provide reporter cells in which to introduce the CAR constructs and measure the activity. We would provide target cells. So in the case of an anti-BCMA CAR, we would provide BCMA expressing cells and BCMA KO cells to make sure that the construct is selective for BCMA. And finally, we would use an anti-BCMA CAR luciferase reporter cell line that the scientists would use as a positive control and an internal control throughout their comparison and their experiments. We would set up for them a validated co-culture assay that uses both these positive control and BCMA expressing target cells. So the recipient cells in this particular case are going to be NFAT luciferase reporter JOCAT cell. The JOCAT cell is a T cell, so it is relevant to the system we're using. And the NFAT luciferase reporter is going to respond to activation by producing luciferase, and then luciferase activity can be measured, and the amount of light that is produced is directly proportional to the activation of the NFAT. So when scientists introduce their CAR construct into this cell, anytime that they activate their CAR, they're going to have luciferase activity directly dependent on the efficacy of the activation of the CAR. So they can compare all of their CAR constructs, see which one is better for T cell activation. Then for them, we'll produce BCMA expressing and BCMA knockout cells. So in this particular case, we have BCMA CHO recombinant cell line. Using the CHO cell line allows to have minimal background. So only the human BCMA is going to be relevant to these experiments. And we have cells that express high levels, medium levels, or low level of BCMA, which may be better reflective of what happens in the clinical situation, where the same patient may have a population of high to low level expressing, or different patients may have different levels of expression of BCMA, and you may want to see if your CAR construct is going to work efficiently against cells that express high levels or against cells that express low levels of BCMA. So this is a flow cytometry analysis of expression showing the control here in red, and then the low level of expression, the medium level of expression in purple, the high level of expression in green. We also have knockout cell lines that we make from BCMA relevant expressing cells. So in this particular case, BCMA is expressed in human MN1S cells. Actually, I think these are murine cells. And then in human RPMI A226 cells, which express high levels of BCMA and are human cancer cells that are relevant to the biology of BCMA. Finally, we will produce a positive control for the cytotoxicity experiments and the activation experiments. We use the recipient cells that I have described a little prior, that are the NF-80 luciferase reporter cells, and we introduce an anti-BCMA CAR into these cells, and we produce a stable cell line that can be used as a positive control. Anytime these cells are in contact with a BCMA expressive cells, they're going to be activated. So the first step of validation, of course, is to verify the expression of the CAR. For these, we use different tools. We can use a recombinant protein that is fluorescent labeled. We can use a recombinant protein that is biotin labeled and can be recognized by an avidin HRP, for example. Sometimes we use a protein that has a tag that can be recognized by an antibody. In this particular case, shown in this flow cytometry experiment, we have used a P-labeled recombinant BCMA protein. So this protein is recognized by the CAR, and that allows us to measure the expression of the CAR on the cell surface. So in purple is the control, in green is the CAR expressing cell line that we have obtained. And the final step for the scientists that requested our help would be to set up the co-culture experiment, in which we add the cancer cell with a target protein. In this particular case, we add the BCMA expressing cancer cells, and we add the anti-BCMA CAR cell line. Once they're added together, we see a production of luciferase that can be measured through luciferase activities. So I've shown two examples in this particular slide. So one is the co-culture assay of the anti-BCMA CAR with soluble BCMA, which is shown here, so there's no activation, or with BCMA CHO cells, which is shown here, and we see a 20-fold induction of luciferase production, luciferase activity in these cells, which show very robust activation of the CAR in this situation. We also have an anti-CD19 CAR cell line and CD19 expressing CHO cells, and we see again very robust activation of the CAR in a co-culture system in which the cancer cell expresses CD19. So now we're going through another case study in which the scientist has moved on to functional validation of his CAR T cells. So these scientists already have their chosen CAR constructs. They know which one they want to use, and they want to validate it further to measure cellular cytotoxicity. So they want to measure cell killing in a co-culture assay, which really reflects the effector function of their T cell. And they want to evaluate the selectivity of their T cell using in vivo models. So in this particular case, they'll use CDX model. BPS Bioscience will provide the wild type and the KO luciferase cancer cells for seamless assessment of target cell death in vitro and of tumor growth in vivo. So in this particular case, the advantage of using a luciferase target cell is that luciferase activity is going to correlate with the number of live target cells without any interference from the immune cells. When you're trying to measure cell killing or the number of live cells, the difficulty can be that you also have the immune cells, which are also live cells. So if you're using a system that's not specific of the target cells, so your signal has a lot of noise because of the presence of the CAR T cells. In these particular cells, if only the target cells are luminescent, the luminescent signal is only due to the target cells. So you have your CAR T cell. In this case, of course, we don't use the luminescent CAR T cells. We use a normal CAR T cells, for example, primary immune cells, in which we would introduce the CAR construct, and the target cells is luminescent. So when at the start of the co-culture, you have maximal signal, and then as the cells are killed, your signal decreases. So we have used the BCMA luciferase CHO cell line in this case. You can see that these cells have strong expression of BCMA. Without lysis, the cells show a very strong signal, and then when you add the CAR T cells and they kill the target cells, you have no signal at all. These cells can then be used as saline xenograft models. So in this particular cell case, you would grow the luciferase expressing cell line. You would inject it into mice for tumor engraftment using immunodeficient mice. And then you can do bioluminescence imaging, where you see that the number of cells in the tumor produces luciferase activity that can be measured with a bioluminescence system. So the signal is directly proportional to the number of luminescent cells in your tumor cells. And when you add your CAR T cell, you can see the killing of your tumor cells. These cells are verified to be mycoplasma free. I don't know why this is here, but it's not me doing this movement. So I'm sorry, ignore the red dot here and just look at my arrow here. So these cells are verified to be mycoplasma free. They have uniform expression of luciferase, so that allows for consistent readouts during the in vivo experiments, and they're very affordable. We have well-established, well-characterized human tumor cells that express BCMA or express CD19. For example, for BCMA, we have the RPMI A226, and then we have the MM1S. And selectivity studies can be facilitated by our KO luciferase cells. I have shown here 3 knockout cells. We also have cells that have GFP that can be used for fluorescence studies. So very useful in vivo studies or in vitro studies. Now let's move on to the TCR-T cells. I'm going to describe a little bit what the T-cell receptor is, what we would use as recipient cells when we want to introduce TCR constructs, what we use for functional validation in co-culture assays, and I'll talk very briefly about primary T cells. So the T-cell receptor recognizes antigens that are presented by the MHC complex, which is a major histocompatibility complex one. So we see here the situation of an infected or an abnormal cell that is presenting a little peptide that has been produced from inside the cell. The TCR is going to recognize this peptide if it is a non-self peptide, so if it is exogenous to the person, and the TCR will interact with the CD3 and with the CD8 to activate the T cell to produce an immune response. So this whole complex is responsible for the activation signals. These activation signals include proliferation of the T cell, production of cytokines, production of enzymes such as granzymes that are responsible for killing the infected or the abnormal cell. So in TCR-T cells, this TCR portion here is replaced with an engineered TCR that has been identified to recognize a very specific antigen of interest. So in this situation, what you do is that you make your construct encoding your TCR, introduce it into a viral vector. You transduce your T cells with your new TCR, and this particular TCR will interact with a peptide that is specifically presented by tumor cells so that you can activate your T cell and induce cell death. Your antigen-specific TCR that you have introduced, if it's built properly, will complex with the endogenous CD3 in the T cell and will function like a normal TCR. So current antigens of interest in the field include PRAME, MAGE, MART1, NY-ESO, and KRAS G12D. That's a typo here. It should be a G. So G12D, which is a mutated form of KRAS. For early phase experiments, TCR knockout cells are very often used as recipient cells. So this will be what I describe here. So these TCR knockout reporter cells are used to express an exogenous antigen-specific TCR molecule for functional validation. So we have established TCR knockout cells. These cells still have the CD3. They have an NFAT responsive reporter, luciferase reporter gene that allows production of luciferase when the NFAT is activated. So we offer CD4 plus TCR knockout, NFAT luciferase cells, CD8 plus cells, TCR knockout cells that do not have a reporter. This can be used in functional experiments in which it is the target cells that has the luciferase activity, for example. We have TCR knockout luciferase reporter, and we also have B2M knockout cell lines for scientists who are more interested in looking at HLA, different HLA alleles, for example. So these cells exist as TCR B2M knockout without the luciferase, or TCR B2M knockout with the NFAT conditional luciferase reporter. So a lot of choices as recipient cells. What we can see here is a PE conjugated anti-TCR antibody to show that the TCR has been fully knocked out. That in green is the knockout cell, in blue is the parental cell. And this shows luciferase activity that is induced using an anti-CD3 agonist antibody. That's an antibody that activates the CD3 in the T cells. And we can see here that the NFAT luciferase, the original cells, do have strong luciferase activity when you activate the CD3, but the TCR-KO cells show no activation anymore because the TCR is not there anymore. So these cells are used to reintroduce a TCR of interest. And as was the case with the CAR cells, we also have a number of cells that do express a specific TCR that scientists can use as positive control. So in this particular case, we can express TCR of interest, and we can validate these new cell lines by measuring the activation upon target binding. The target cells here are antigen-presenting cells in which we have loaded the peptide of interest. So for example, we can use a MART1 peptide, or we can use an NY-ESO peptide. This peptide will be presented by the MHC1 at the surface of the antigen-presented cell. And when we put the cells together, the antigen-presenting cells and the TCR-expressing cells, we have activation of this exogenous TCR, which results in production of light because of the luciferase activity. So you can use these cells as positive control when you're designing your own TCR. I'm showing here the example of a CD8 plus TCR-KO cell. So in this one, in this control, the TCR has not been reintroduced. So these cells have no TCR, and they cannot be activated by antigen-presenting cells, whether it's NY-ESO or whether it's SMART. We have an AY-ESO TCR-specific cell line. These cells are activated by antigen-presenting cells that have been preloaded with the NY-ESO peptide, as shown here. And it is very specific because when you're adding the MART peptide to these cells, you see no activation of the NY-ESO TCR. Conversely, if you're using the MART1 TCR, you have no activation by cells that have been loaded with the NY-ESO peptide here, but you do have activation when you're co-culturing the cells with antigen presenting cells that have been loaded with the MART1 peptide. And finally, we're using primary cells to evaluate the CAR or the TCR constructs. These cells are used when you are at a stage when you want a more biologically relevant immune cell population. So the immune cells are purified from peripheral blood mononuclear cells, and they are transduced with a CAR construct of interest or a TCR construct of interest. And it can be used as positive control in experiments that are involving human T cells. This is the workflow here from the collection of peripheral blood, screening for pathogens, because these are now coming from humans, the collection of the PBMC from the blood draw, and then the magnetic separation of CD4 plus or CD8 plus primary cells, in which we can introduce CAR or TCR. So we have pre-made CAR T cells, pre-made TCR T cells that we offer, so that you don't have to go through that whole process. You can just buy these cells. And then we have introduced controlled T cells that you can use as negative control in your cells and in your experiments. So just a quick view of our custom services for CAR T cells and TCR T cells. We do recognize that buying a cell line can be expensive if you're not sure that they're going to be working for you. So we have a cell line rental program where you can try the cells before you actually buy them. The option is available for more than 150 different cell lines. You can rent for up to 3 months for 50% of the actual price. And at the end of the rental period, if the cells are working for you, you can just purchase with the rest of the 50%. So you can buy target cells, you can buy your control CAR T cells, your control TCR T cells, and so forth. You can rent them and try them before you actually purchase the cell line. We also offer full custom CAR services or TCR services. This is a milestone measure process in which you have results of experiments after every step of the process. So the researcher would provide us with the antibody sequence against the antigen. We will make the construct. We will build the lentivirus or the AAV virus, depending on what you want to do. We'll prepare T cells. We'll proceed with the functional validation of the CAR or the TCR T cells, and we will provide T cell expansion and T cell optimization. So every step is available to scientists who need them. Finally, I would like to show you a couple of posters that we have presented at meetings. So one is really describing all of the tools that we have for TCR T-cell research, including a few tools that are very new to BPS that I haven't talked about, that are used to study HLA, different alleles. We are developing currently HLA negative cells. So these are a whole new product line that we will be working on this year. And we also have a new product line on gamma delta T cell. This is still in development, but we're starting to offering products. So if you want to learn more about the gamma delta T cells, please go look to this one poster. We have a poster on bispecific molecules and characterization of bispecific molecules, including dual antigen bridging ELISA-KIT, characterization strategies. And finally, I'm putting it here, we have a poster on incretin agonists like GLP-1R, GLPR, and GCGR. luciferase assays for development of GLP-1 agonist. And I think that's it for me today. So thank you very much, and I will take any question you may have.
Exactly. Thank you so much again, Veronique. So to wrap up the session, let's move on to the Q&A session. Just here, changing the slide. I have received a couple of ones. So what is the advantage of using a luciferase reporter cell line, like the cells you discussed, when we perform a co-culture experiment?
Right, so it depends a little bit on which one system you're using. So in one system where I have spoken about the target cells, so in the case where the target cells are luciferase positive, then you can really make the differentiation between the immune cell in the co-culture and the target cells. The immune cells don't have the luciferase, the target cells do. And so the luciferase signal is going to be strictly dependent on the amount of live target cells that are there. So if you're measuring cell killing, your signal will decrease and only it reflects only the death of the target cells. Now, if you're using a luciferase that is in the CAR T cell or the TCR-T cell, what this allows is measuring the activation of the construct. And the advantage here is that it's very specific of the activation of your CAR or your TCR. It's quantitative. That's the main advantage. You can compare different constructs by measuring the EC50 of your construct. So it's very dose dependent, right? If you add an increasing number of target cells, you're going to have an increasing signal. If your CAR is more effective in one conformation compared to the other, your EC50 is going to move. So you can really compare different constructs. It's very quantitative, very sensitive. It's a relatively stable signal, and you can make all the measurements that you need to make to decide which construct is the best one for you.
Thank you again. There is another one. Are the anti-BCMA CAR NFAT luciferase is a CAR or of what generation?
So if I remember correctly, we're now offering two different BCMA CAR and two different CD19, and I think one of them is third generation and one of them is fourth generation. I would have to check on that really. Yeah, but I think that's what it is.
Okay, and there is something else with the question. Are different generations of cars available? So I guess yes.
So I think the answer is yes, and then again, we can make new ones. So if a scientist is interested in having a different situation, a different generation, we're happy to make it for them.
Okay. And to finish with this one, do you sell NFAT luciferase DURCAT cell not expressing any CAR?
Yes, so these are the recipient cells, what I have been calling the recipient cells that scientists use to introduce their own CAR. So in that particular case, you can even use a TCR KO cell if you want to do this. So our NFAT luciferase DURCAT cells exist in many versions. You just have the NFAT luciferase DURCAT cell, you have the TCR KO NFAT luciferase, you have the CAR NFAT luciferase. So you can use different versions as you want. The NFAT luciferase, for example, can be used to introduce a CAR. The TCR KO ones are better to introduce a new TCR. And of course, the different positive controls are best to, when you're at the stage where you want to set up your co-culture assay and you want to optimize it and set up in your lab, you would want something that you know works. So in that case, you would use a CAR expressing or a TCR expressing. But yes, they do exist without CAR expression.
Okay, thank you. And I think I have another one. if I work with a distributor, will I still be able to have access to BPS scientists to discuss my work?
Absolutely. So we are always happy to answer any question. In tricky situations where you really need to talk to a scientist, what we would do is, I think we'd have a meeting with people from TB Bio and one or two of our scientists where we would discuss the project and we would discuss what the best tool would be for this project, and how we can help at any stage of the project.
Perfect. Thank you again. So far, I don't have any further question or that we have received. Oh yes, I have one. Can the NF80 luciferase reporter JURCAT cell be used for LNP screening?
Can you define LNP for me, or are we talking about
The
like lipid particles?
nanoparticles, yep.
Lipid nanoparticles. Okay. I think so. I mean, I don't see why not. We use them for, for lentivirus transduction. We can use them for testing AAV. So I don't see why we could not use them to test LNP. I suppose what you would want to do in this case is compare different LNP that are used to introduce a new CAR, for example. So that'd be perfectly acceptable, I think.
Okay. Just ask if the person wants to specify in case. And yeah, so if we have not received anything else, let's conclude the session.
Let's
So
wrap up.
once more, I wanted to take a moment. If you could take a moment, please, to complete the survey that will appear in your browser when you're going to leave the session, that would be really great. And again, thank you all for your attention, for joining us today. And we hope the information shared has been really helpful and inspiring. So feel free to reach out if you have any further questions, and if you would like to discuss with us, don't hesitate. Again,
Thank you, Diana.
Dr. Baron, thank you so much, Veronique,
Thank you.
and have a lovely day. That I know that it's a long day for you now, and for us here in Europe, it's almost the end. So thank you again
Have
for
a
your
great
help.
evening.
Okay, bye bye
Bye-bye.
everyone.
Bye.